The arrival of aerosols to the marine environment play an important role in Earth's cycles. However, there are numerous trace metal sources, such as ice melts, riverine inputs, hydrothermalism, and wet deposition. All these elements mix within the water column. Some trace metal examples are copper, nickel, zinc, cobalt, manganese, and iron, of which we will be talking about. Iron in seawater can be found in two oxidation states, iron 2 and the oxidized iron 3. Both oxidation states coexist and are reversible. Iron is an essential micronutrient for life. It is essential for photosynthesis and phytoplankton growth. Primary production plays a pivotal role in carbon dioxide uptake, reducing atmospheric concentrations, mitigating anthropogenic emissions. Phytoplankton are the starting element of most marine food chains. Iron can be complexed by organic compounds. They influence the rate at which iron 2 is oxidized. Iron 2 oxidation rate can be affected by many physicochemical parameters. Oxygen concentration tends to accelerate the oxidation rate. pH decreases slow down oxidation rates. Temperature increases speed up oxidation rates. Light affects the oxidation rate depending on seawater composition. Deep seawater samples can be collected. They are brought to the ocean surface to elucidate the uncertainties of our ecosystem. Oxidation kinetic experiments are performed in trace metal clean conditions under ISO 6 regulation. We follow Geotrace's procedures. Personal protection equipment is used to protect samples from any contamination. Coveralls and laboratory shoes are required. Tacky mats are required to eliminate any particles attached to the shoes. Since oxidation is temperature dependent, a water bath is used to acclimate the samples to the experimental conditions. Experiments are performed inside an ISO 5 laminar flow hood, which is constantly kept clean. To perform the analysis, we use two reagents. First, a buffer, to regulate the reaction pH made from acetic acid and ammonia. The pH is adjusted to pH 5.5. Every time a pipette tip is used, they are previously rinsed with diluted hydrochloric acid and ultra-high purity water. The first reagent aspiration is discarded and used to acclimate the tip. Luminol oxidizes during the oxidation of iron 2. Maximum luminol efficiency is obtained about 1 month after its preparation. We monthly prepared a concentrated stock, and daily we produce a diluted stock. To observe iron 2 oxidation, we first need to add the iron 2. Analyzed samples have to be filtered. Samples are filtered using acid-clean syringes, filters, and filter holders. All the components are rinsed with a sample. We use 50 ml of sample. Which are poured into the reaction vessel. The reaction vessel is inserted into a temperature-controlled environment. Pure air is pumped to achieve oxygen-saturated conditions. The air is cleaned using potassium permanganate and an air filter. Samples are aerated for 20 minutes before analysis. pH is controlled using an automated system. Sample and reagent lines are cleaned using hydrochloric acid and ultra-high purity water. After which, They are introduced into their bottles. A known amount of iron 2 is added into the sample. 50 microliters are used to achieve a 0.7 nanomolar concentration. The sample line is inserted, and the analysis starts. The sample and buffer mix before arriving to the sensor, where it combines with the luminol. The reaction emits a signal which is recorded. The signal decreases as iron 2 concentration decreases.